Okay. Well, thank you again, and good afternoon, everyone. My talk today is just going to be about the treatment of mixed chlorinated solvents and 1,4-dioxane in groundwater samples. So, a little bit of background on 1,4-dioxane is it's an emerging contaminant, and it's typically found at sites that do have chlorinated solvents because its historical uses have typically been as a stabilizer for these chlorinated solvents. Um, it was used because what people found is you had chlorinated solvents that were typically stored in drums, typically metal. And if the chlorinated solvents were stored in there for any extended period of time, the chlorinated solvents could break down, releasing chlorine into the headspace of the drums. Then, therefore, the free chlorine would react with the moisture that would be in the tops of the drums and therefore starting to corrode the drums, hence the need for some sort of a stabilizer. So this stabilizer was used in these drums. The problem with 1,4-dioxane is it's a readily water-soluble material. So we were going to look at two different biodegradation strategies to try to figure out a way that we could remediate this problem at this site. So in this first slide, I've just kind of shown the biodegradation pathways for both 1,4-dioxane and for chlorinated volatile organics. So the pathway for 1,4-dioxane in the literature has typically been a degradation pathway under aerobic conditions. So you can see that the 1,4-dioxane breaks down into various different daughter products, and then in the end can break down into daughter final products like glyoxylate, ethylene glycol, glycolate. And in the case of chlorinated solvents, the typical pathway for those is an anaerobic degradation pathway, typically a dehalogenation type of breakdown using some sort of, dihalococoides is the sort of common bacterial name that is used for degrading chlorinated solvents all the way to an ethene final product. So in our study, see here. Oh, come on. There. Oh, there we go. So this strategy here, again, we ran into, okay, we have chlorinated solvents. They degrade anaerobically. 1,4-dioxane, it degrades aerobically. So we wanted to try to assess the potential of just using an enhanced biodegradation treatment for the chlorinated using an anaerobic degradation, and then using an enhanced biodegradation of 1,4-dioxane using an aerobic. And then looking into to see, well, what if we did both of these processes in the same microcosm, just degrade one compound under one condition and then switch the conditions at some point once we see that compound starting to go away. So typically for anaerobic degradation, you generally set up some microcosms that have different treatments in them. We typically use here in our treatability lab a control sample where we just have a soil and groundwater mixture to see if the natural degradation can have any impact. And then we enhance it by adding such amendments such as nutrients. In the case of anaerobic, it's some sort of carbon source, usually an emulsified vegetable oil or a lactate. And then depending on what we see happening, it may be necessary to add some sort of commercial inoculant.
1 minute remaining.
And then the same goes for the aerobic conditions, which is usually just using the same type of treatments, but adding oxygen to make sure the conditions stay aerobic. So we did this and we kind of saw that the results showed that we can remove chlorinateds under anaerobic conditions very well. We did see that the 1,4-dioxane can also be degraded under aerobic conditions. However, since the greater treatment of both the 1,4-dioxane and the chlorinated were observed with actually a sequential treatment, therefore using both aerobic and anaerobic, we decided to suggest this kind of sort of co-metabolic treatment for the field study at the site. And that is all. Thank you.